Okay, so the last few videos have kind of talked more about the evolutionary side, talking about fitness, adaptations, physical constraints and traits, ideas like comparative anatomy, and a bit about what scientific theories are, and a bit about Darwin and his ideas on evolution, and how he came to those ideas. Now we're going to move to a bit more on the genetic side of things, and how we actually discovered that DNA was the genetic material that actually is responsible for causing these traits that Darwin had observed. So linking the genetic side of things to the evolutionary side of things done by these original naturalists like Darwin. So this kind of links into the adaptation of Darwinian theory in the early 20th century, when it was integrated with Mendel and other kind of ideas to produce the modern synthesis of kind of evolution, which then would be adapted again towards the end of this playlist to produce the kind of Evo-Devo kind of school for homeobox genes. So let's just get into this. For some time, it was debated whether chromosomes were the genetic material or just involved in the transmission of the genetic material. I don't know what that was. It was originally thought that proteins were the genetic material, while DNA was merely a sugary structure from which these proteins would hang. This was obviously not the case. As we know today, DNA is the structure responsible for producing these traits that we've been talking about. Each of these traits are coded by genes, and they can either be monogenic, controlled by single genes, or polygenic, controlled by many genes, which we go over in some of the future videos on quantitative traits. So, an experiment was done in 1928 by Frederick Griffith, who took a harmless strain of Streptococcus pneumonia and mixed the harmless strain with a heat-killed deadly pathogenic strain. The mixture of this harmless and deadly strain was injected into mice, and they promptly died. This was because some of the plasmids from the deadly strain were transferred to the harmless strain via conjugation, making the harmless strain deadly. This principle showed us that genetic information was transferred by plasmids in prokaryotes. The Avery-McLeod-McCarty experiment, which we're going to go over next, was the experiment that proved that DNA itself was the genetic material. So, let's get into the Avery-McLeod and McCarty experiment. The experiment basically showed that DNA was the genetic material by giving a bacteriophage, shown on the right, the radioactive isotopes P32 and S35, so phosphorus 32 and sulfur 35, which each emit different levels of radiation. The bacteriophage inserts the genetic material into the target cell to make it synthesize more bacteriophage DNA and other essential proteins required for making more bacteriophages. The scientists then measured the radioactivity of the target cell to determine whether the bacteriophage had incorporated the P32, which was part of the bacteriophage DNA, or whether the bacteriophage had inserted the S35 isotopes, which would have been associated with bacteriophage proteins. So they basically checked the radioactivity of the target cells to see whether the bacteriophage had inserted DNA into the target cell, or if it had inserted proteins into the target cell. And when they did this, they found out that the bacteriophage had inserted DNA into the target cell, indicated by the cell having radioactivity from the P32 isotopes, which would have been part of the sugar phosphate backbone of DNA. Meanwhile, the sulfur would have been part of proteins, because obviously the amino acids methionine and cysteine have sulfur incorporated into their structure. So, moving on to the Mattei and Nuremberg experiment. Basically, the genetic code is read in sequences of 3 nucleotides called codons. So you can see this image on the right that I've made here. UUU dash UUU dash UUU dash UUU. This UUU code is a codon, so the 3 bases of uracil are a codon. They're read at a ribosome, and the ribosome then recognizes that codon, and a tDNA, which we're not going to go into, but a tDNA brings the amino acid which that codon codes for to the ribosome, and it's added to the polypeptide chain. So you can see here it's reading a bunch of codons for phenylalanine. So at the ribosome here, they're adding phenylalanine amino acids, and they put all these together to produce a polypeptide with all these amino acids produced by the codons. There have to be 3 nucleotides minimum per codon to produce all 20 amino acids, because 2 nucleotides per codon could only produce 16 amino acids, while 3 is able to code for 64 amino acids, which is more than enough, which is why if you look at the amino acid chart or table, a lot of amino acids are coded for by several different combinations of codons, just because we have more potential codon variations than amino acids available. So in 1961, Mattei and Nuremberg put polynucleotide fragments with known repeated DNA sequences into a suspension of E. coli cytoplasm containing ribosomes and other essential organelles. So the ribosome being this structure here. They put these in 20 test tubes, each containing all 20 amino acids, and they would radioactively label one of these 20 amino acids. They would then determine which test tube had the radioactively labeled amino acids joined together to form a polypeptide, and they would then they would know what triplet codon of the polynucleotide fragment would code for the amino acid. So going back to this example that I produced on the right, this would be the polynucleotide fragment over here, each with these sections of 3 bases called the codons. Each codon codes for the same amino acid here because all the codons are exactly the same. This would be mRNA. The mRNA would be taken to a ribosome and translated, and this would be a methionine amino acid, this would be leucine. In this example, these are one of the 19 amino acids that are not radioactively labeled, where this one is the radioactively labeled amino acid for this Mattei and Nuremberg experiment, phenylalanine. They would know that the mRNA is made of all of these codons, which are all exactly the same. So when they would insert it together, when they put these amino acids together during the translation process to produce a polypeptide, it becomes heavier, they collect at the bottom, so they're basically looking for which amino acids are being put together to form a polypeptide from the polynucleotide fragment that they're using.